Hello everyone. So today is the fourth problem solving session for the course Overview and Integration of Cellular Metabolism. This is an NPTEL course. The course instructors are Professor Ritri Beer and Professor Indam Ghosh. And I'm Shrija Ghosh of PMRF from IIT Kanpur. I'm here to help you all to solve the assignment questions related to each week's topic. So today's topic is all about galactose metabolism, fructose metabolism, and then how glucose is regulated in our body and uh, how abnormality in galactose and fructose mechanism leads to certain diseases, certain disorders. Then we'll learn about diabetes mellitus, and then finally we'll start with the lipid absorption, how the lipids are broken down and absorbed in our body. Okay, so I have certain questions related to these topics, and if you have any doubt in any of the questions, you can ask me. uh, I'll start with the questions, and at any feel free to ask me anything at any point. I'll keep explaining the concepts related to those questions, uh, which will help you solve the assignment questions for this week. Okay, so I'll start with the first question. Question number one. Galactose is converted to glucose 1-phosphate through a series of enzymatic reaction. Which enzyme is deficient in classic galactosemia? Galactose is converted to glucose 1-phosphate through a series of enzymatic reaction. Which enzyme is deficient in classic galactosemia? Option A, galactokinase. Option B, galactose 1-phosphate uridylyl transferase. Option C, galactose 6-phosphate epimerase. And option D, aldolase B. So, galactose is converted to glucose 1-phosphate. Which enzyme is deficient in classic galactosemia? So, the option A is galactokinase, option B galactose 1-phosphate uridylyl transferase, option C galactose 6-phosphate epimerase, and option D aldolase B. If you know the correct answer, you can write it in the chat box. So the correct answer is option B. Uh, Deficiency of galactose 1-phosphate uridyl transferase leads to classic galactosemia. So, basically, this is the galactose metabolism process. So, it starts from galactose. Galactose is first phosphorylated using galactokinase. I told you in the previous sessions also, wherever kinase is present, that means there is a transfer of phosphate group. So, in this case, galactose is phosphorylated by this enzyme galactokinase and it becomes galactose 1-phosphate. This galactose 1-phosphate is then converted to UDP galactose with the help of enzyme galactose 1-phosphate uridyl transferase and a UDP glucose molecule. So, if you remember this UDP glucose, we studied this UDP glucose in the previous class. This was the active form of glucose for the glycogen synthesis, right? So, for glycogenesis or for glycogen synthesis, we need glucose units, right? Glycogen is a storage unit of glucose. So these glucose are activated by addition of UDP. So UDP glucose is the active form. This then basically proceeds to form glycogen. Okay, so this UDP glucose combines with galactose 1 phosphate with the help of enzyme galactose 1 phosphate uridyl transferase, and the UDP from the UDP glucose is transferred to the galactose 1 phosphate. So so the product we'll get is UDP galactose and a glucose 1 phosphate. So you see the UDP from UDP glucose is transferred to galactose, and the 1 phosphate group that was there with galactose, that is transferred to the glucose. So now we have glucose 1 phosphate and UDP galactose. Okay, so two products are there, and this UDP galactose can be converted to UDP glucose by epimerase. Since these two are epimers of each other, these two are chemically same. Like if you see the number of carbon, hydrogen, oxygen, it's the same molecule, just the conformation is different. So the major difference is in this fourth OH group, fourth carbon's OH group. One is on the left hand side and one is on the right hand side in a Fisher projection. So UDP galactose is an epimer of UDP glucose, and it can be converted to UDP glucose by the enzyme UDP galactose 4 epimerase. 4 is because it's the fourth carbon. So this is the first carbon, second carbon, third carbon, and then fourth carbon. Okay, so 4 epimerase. In the fourth position, the epimerization occurs. So UDP galactose becomes UDP glucose, and this UDP glucose then further can drive the process in the forward direction. So now what happens in classic galactosemia? This enzyme is deficient. What will happen if this enzyme is deficient? If this enzyme is not there, that means this reaction cannot proceed, and there will be an accumulation of galactose 1 phosphate in the body. Okay, so in classic galactosemia, it's a deficiency of galactose 1 phosphate uridyl transferase. Okay, so since this enzyme is not there, now UDP can't be transferred from glucose to galactose, and galactose 1 phosphate will keep getting accumulated because the reaction can't go further ahead. Okay, I hope that's clear. So galactosemia is basically deficiency of galactose 1 phosphate uridyl transferase. Moving to the next question. Which of the following is not a characteristic feature of classic galactosemia? Option A, hyperglycemia. Option B, hypoglycemia. Option C, unconjugated hyperbilirubinemia. And option D, galactoseuria. Which of the following is not a characteristic feature of classic galactosemia? Which of the following is not a characteristic feature of classic galactosemia? Hyperglycemia, hypoglycemia, unconjugated hyperbilirubinemia, and galactoseuria. So what is the correct answer? You can write your answers in the chat box. Do you know the correct answer? Yes, hyperglycemia, that is the correct answer. So option A, hyperglycemia is not a feature of classic galactosemia. During galactosemia, we actually have hypoglycemia. Okay, and all these other features are also related to associated to galactosemia. So as I've mentioned in the previous question, galactosemia occurs due to deficiency of the enzyme galactose 1 phosphate uridyl transferase. So in the galactose metabolism, galactose first gets converted to galactose 1 phosphate using galactokinase, and then uh, galactose 1 phosphate uridyl transferase can transfer one UDP molecule from the UDP glucose to the galactose, and the phosphate is transferred to the glucose. So as a result, we get UDP galactose plus glucose 1 phosphate. And in galactosemia, this enzyme is absent. Due to absence of this enzyme, galactose 1 phosphate keeps getting accumulated. So due to the block in this enzyme, galactose 1 phosphate will accumulate in the liver. This will inhibit galactokinase as well as glycogen phosphorylase. So basically, as I told you in the previous slide, we have galactose that is converted to galactose 1 phosphate. Now, if galactose 1 phosphate is getting accumulated, it kind of gives the signal to the galactose kinase to stop producing to stop metabolizing the galactose. So in that case, galactose will also keep getting accumulated. So galactokinase is basically inhibited. It has a product inhibitory effect. So since galactose 1 phosphate keeps on increasing, uh, it will inhibit the galactokinase. It will signal the galactokinase that do not break down or do not convert more and more galactose into galactose 1 phosphate, because we have enough concentration. Now that will lead to accumulation of galactose. Okay, and it also inhibits glycogen phosphorylase. So if from the previous session, if you remember, glycogen phosphorylase was the enzyme responsible for the glycogen breakdown. Okay, so glycogenolysis. So glycogenolysis or glycogen breakdown results in the formation of glucose. Now if it is inhibiting glycogen phosphorylase, it is technically inhibiting the glycogenolysis process or glycogen breakdown process that will ultimately lead to hypoglycemia or less or low blood glucose. Okay, so it results in neonatal hypoglycemia, and galactose cannot be converted to glucose. uh, so this galactose that is there, that also cannot be converted to glucose, as you can see. And increased galactose level increases insulin secretion, which lowers blood glucose level. So high galactose signals the insulin secretion, and insulin secretion, triggers the glucose metabolism. So whatever glucose is present in the cell, they will go through glycolytic processes. So insulin secretion will lower the blood glucose level further. So this will lead to hypoglycemia. Okay, so hypoglycemia is a characteristic feature of galactosemia, not hyperglycemia. Okay, I hope that's clear. If you have any doubt, you can write it in the chat box. Okay, I'll move on to the next question. Which disorder is characterized by a deficiency of the enzyme fructose 1 phosphate aldolase, that is aldolase B? Which disorder is characterized by a deficiency of the enzyme fructose 1 phosphate aldolase, that is aldolase B? Option A, fructose malabsorption. Option B, essential fructosuria. Option C, hereditary fructose intolerance. Option D, fructose 1,6 bisphosphatase deficiency. Which disorder is characterized by deficiency of the enzyme fructose 1 phosphate aldolase? You are saying option C. that is hereditary fructose intolerance. That is the correct answer. So deficiency of aldolase B leads to hereditary fructose intolerance. So during fructose metabolism, like in galactose metabolism, we saw galactose is first converted to galactose 1-phosphate. Similarly, in fructose metabolism also, fructose is first converted to fructose 1-phosphate by fructokinase. Then this fructose 1-phosphate gets converted to dihydroxyacetone phosphate and glyceraldehyde. So if you remember from glycolysis, we had a similar reaction with an aldolase enzyme, that was aldolase A. So in that also we had a 6 carbon sugar that was broken down to 2 3 carbon sugars. So in that there we had fructose 1,6 bisphosphate getting converted to dihydroxyacetone phosphate and glyceraldehyde 3 phosphate, right? So here similarly, since we have only one phosphate group, so it is broken down into dihydroxyacetone phosphate and glyceraldehyde. Later this glyceraldehyde can be phosphorylated by a kinase to form glyceraldehyde 3 phosphate, and these two metabolites can directly enter the glycolytic pathway. So, so this dihydroxyacetone phosphate and glyceraldehyde 3 phosphate, they both can directly enter the glycolysis or glycolytic pathway. Okay. So in hereditary fructose intolerance, this aldolase B is absent. Okay. So due to deficiency of aldolase B, what will happen? Fructose 1-phosphate cannot get converted into these glyceraldehyde and dihydroxyacetone phosphate, and it will lead to accumulation of fructose 1-phosphate in the system. So fructose 1-phosphate accumulation inhibits the glycogen phosphorylase. So similar to what you saw in galactose metabolism, where galactose 1-phosphate was inhibiting the glycogen phosphorylase, fructose 1-phosphate also inhibits the glycogen phosphorylase. That will again lead to inhibition of the glycogen breakdown or glycogenolysis, ultimately leading to low blood sugar or hypoglycemia. So this results in glycogen accumulation. So glycogen will get accumulated because it's not getting broken down. So glycogen accumulation in the liver and concomitant hypoglycemia. Infants frequently fail to thrive, characterized by vomiting, loss of appetite, hepatomegaly, and jaundice. Hepatomegaly means enlargement of liver. If liver injury advances, cirrhosis and death will ensue. This is seen when sucrose that contains fructose. So sucrose, that is the common sugar that we take, table sugar. Sucrose has two units, fructose and glucose. So sucrose is made of glucose and fructose. Okay. So fructose is present in sucrose, and this is in when sucrose is introduced in the diets of these infants, usually around 6 months of age, then it is seen like kids who don't have this aldolase B and who suffer from hereditary fructose intolerance, uh, it is identified only when fructose is introduced to the body. So unless fructose is there, it is very difficult to identify whether the uh, child has aldolase B deficiency or not. Okay. Then fructose elimination from the diet will promptly alleviate the symptoms. So if you don't uh, provide any source of fructose to the child, then it will alleviate the symptoms. The symptoms will be gone because mainly fructose can't be metabolized. So if you don't give fructose, the problems won't be there. Fructose is excreted in urine that can be tested by Benedict's test or Selivanov's tests. So since fructose 1-phosphate is getting accumulated, that will in turn inhibit the fructokinase after a point, and then fructose will also keep getting accumulated, and that will lead to fructosuria or presence of fructose in urine. So when some metabolites keep getting accumulated in the body, after a point what the body does, it keeps excreting it out in urine. Okay, so that's the body's excretion process. So that's how, so in case of galactosemia also, you have a symptom called galactosuria, that is basically presence of galactose in urine. So galactose, since galactose keep getting accumulated in the body, it will be excreted in the urine. So similarly in this case, fructose is excreted in the urine. Okay, this is clear. So hereditary fructose intolerance basically is the deficiency of aldolase B. What aldolase B does, it breaks down fructose 1-phosphate into two 3 carbon sugars. One is dihydroxyacetone phosphate and one is glyceraldehyde, which can further be converted to glyceraldehyde 3-phosphate, and these two metabolites can directly enter the glycolytic pathway. We have already discussed glycolysis, so I hope this is clear to you. If you have any doubt, you can let me know. Okay. Okay, moving on to the next question now. Which of the following symptoms, which of the following are symptoms of hereditary fructose intolerance? We have already discussed this now. Which of the following are the symptoms of hereditary fructose intolerance? Multiple answers are correct. Option A, hypoglycemia. Option B, hyperglycemia. Option C, fructosuria. And option D, hypertension. What all are the symptoms of hereditary fructose intolerance? We just discussed hereditary fructose intolerance, so you should be able to answer hypoglycemia, hyperglycemia, fructosuria, and hypertension. So you are saying option A and C, hypoglycemia and fructosuria. Yes, that is the correct answer. So we just discussed, due to the absence of aldolase B, hereditary fructose intolerance, in hereditary fructose intolerance, fructose 1-phosphate, or ultimately fructose, keeps getting accumulated. That fructose 1-phosphate in turn inhibits the glycogen phosphorylase. So in as a result, glycogen keeps getting accumulated in the liver and results in hypoglycemia, that is low blood sugar, and fructose keeps getting accumulated, so it is excreted out in the urine, leading to fructosuria. Okay. Next question. Why is the rate of fructose metabolism faster than the glucose metabolism? Why is the rate of fructose metabolism faster than the rate of glucose metabolism? Option A, fructose enters glycolysis before glucose. Option B, fructose is phosphorylated by hexokinase in the liver. Option C, fructose bypasses phosphofructokinase 1 regulation. Option D, fructose needs insulin for transport into cells. Why is the rate of fructose metabolism faster than the glucose metabolism? Option A, fructose enters glycolysis before glucose. Option B, fructose is phosphorylated by hexokinase in the liver. Option C, fructose bypasses the PFK1 regulation. And option D, fructose needs insulin for transport into the cells. What is the correct answer? You are saying option C, that is fructose bypasses the phosphofructokinase 1 PFK1 regulation. That is the correct answer. So option C is the correct answer. So basically, if you remember from glycolysis, we had the glycolytic pathway was starting from glucose, where glucose was getting converted to glucose 6-phosphate, then fructose 6-phosphate, and then fructose 1,6 bisphosphate. So fructose 6-phosphate to fructose 1,6 bisphosphate reaction is catalyzed by the enzyme phosphofructokinase, as you can see here. So this phosphofructokinase basically is a regulatory enzyme, and it is inhibited by certain factors. That is how the glycolysis pathway is regulated. So when enough end products are present, it will inhibit the phosphofructokinase, and then body will get the signal that okay, we don't need to do any more glycolysis. So major feedback inhibition is by the ATP and citrate. So citrate, you know, is a product of TCA cycle, and ATP is a end product of glycolysis. So glycolysis will lead to pyruvate. Pyruvate will form acetyl coenzyme that will enter the TCA cycle, and finally all the electron carriers, everything will enter the electron transport chain, resulting in enormous amount of ATP. That ATP is the ultimate goal of glycolysis, right? So once the body has enough ATP, or once the body has enough citrate, those molecules are going to inhibit phosphofructokinase so that the glycolytic pathway does not proceed anymore. Okay. Another inhibition that occurs is from glucagon. So glucagon, you know, is a hormone that signals the formation of glucose. Okay, insulin is the hormone that signals the breakdown of glucose, whereas glucagon is the hormone that signals the secretion of glucose. So if glucagon is present, then glycolysis will stop and gluconeogenesis will start occurring. So glucagon also kind of inhibits the phosphofructokinase, and then another enzyme that is there, fructose 1,6 bisphosphatase, that is the enzyme that is present in uh, case of gluconeogenesis. So this is a irreversible enzyme. Phosphofructokinase step is a irreversible reaction, right? So fructose 6 phosphate can get converted to fructose 1,6 bisphosphate by this kinase enzyme. And for the reverse reaction, for the dephosphorylation, we need fructose 1,6 bisphosphatase. So that enzyme automatically inhibits the phosphofructokinase. Okay, and insulin uh, activates this step. Okay, so now all these feedback inhibition steps are happening before this process, before the acetone phosphate and glyceraldehyde 3 phosphate formation, right? So all the feedback inhibition is happening before this process. But if you uh, remember from the previous slide, the fructose metabolism, uh, in the fructose metabolism, fructose is getting converted to fructose 1 phosphate by fructokinase, and then fructose 1 phosphate is getting converted to glyceraldehyde and dihydroxyacetone phosphate, which are entering the glycolytic pathway. And this point is after the uh, checkpoint of glycolysis. So that's why fructose metabolism is faster than the glucose metabolism, because glucose metabolism is kind of controlled, it's kind of regulated, but that regulation is now not present in the fructose metabolism. So there are two irreversible steps in glycolysis. One is this hexokinase or glucokinase step, and one is this phosphofructokinase step, and both of these steps are skipped in case of fructose metabolism because it is entering the pathway at this point, and at this point there are no checks to regulate the mechanism, and then more and more acetyl coenzyme A will keep forming because nothing is inhibiting this part. Do you understand? So rate of fructose metabolism is going to be faster than rate of glucose metabolism because there's no regulation in the mechanism, in the metabolic pathway. Is that clear? And then ultimately more and more acetyl coenzyme A get if more and more acetyl coenzyme A keeps getting accumulated, what will happen? Not all the acetyl coenzyme A can enter the TCA cycle because oxaloacetate and other TCA cycle metabolites are going to get depleted. So those acetyl coenzyme A then ultimately go towards the fatty acid synthesis, basically lipid metabolic pathway, which we'll study in the next classes in detail. So that will lead to fat accumulation basically. So more acetyl coenzyme A is also harmful for our body. Okay. I hope that's clear. I'll move on to the next question. Which of the following statement is true? Option A, liver has glucokinase and hexokinase, while most extrahepatic tissue have only glucokinase. Option B, liver has hexokinase, while most extrahepatic tissue have glucokinase and hexokinase. Option C, liver has only glucokinase, while most extrahepatic tissue have both glucokinase and hexokinase. Option D, liver has glucokinase and hexokinase, while most extrahepatic tissue have only hexokinase. Which of the following statement is true? Liver has both glucokinase and hexokinase, while most of the extrahepatic tissue have only glucokinase. Liver has only hexokinase, while most other tissues have glucokinase and hexokinase. Liver has only glucokinase, while most extrahepatic tissue have glucokinase and hexokinase, or liver has glucokinase and hexokinase, while most extrahepatic tissue have only hexokinase. What would be the correct answer? You have mentioned option D. That is, liver has glucokinase and hexokinase, while most extrahepatic tissue have only hexokinase. That is the correct answer. Very good. So, liver basically has both the enzymes. So, if you remember, hexokinase is the enzyme that converts glucose to glucose 6 phosphate in glycolysis, right? So, we have glucose going to glucose 6 phosphate by the enzyme hexokinase or glucokinase. Both can convert glucose to glucose 6 phosphate. Now, this glucokinase and hexokinase, both the enzymes are present in liver, but in other tissue, in muscles and all, only hexokinase is present. Why? Because now this hexokinase is a saturable enzyme. So, saturable means this enzyme will get saturated after a point. So, if a lot of glucose is present, in that case, after a point, hexokinase won't be able to uh, do the phosphorylation of the glucose. Okay, it will get saturated, but glucokinase is not saturable. So even if more and more glucose is present in the liver will keep uh, phosphorylating the glucose to glucose 6 phosphate. Why? Because if you remember, if more glucose is present in the liver, what happens? It goes towards the glycogenesis, right? It goes towards the glycogen synthesis. So if only hexokinase was present in the liver, liver also won't be able to process the remaining glucose. So glucokinase, since it is non-saturable, it will keep converting glucose to glucose 6 phosphate that can enter the glycogenesis pathway. So if you remember, glycogenesis pathway, glycogen synthesis also requires glucose 6 phosphate. Okay, so this for this phosphorylation to occur, uh, liver needs the glucokinase. Liver needs an enzyme that is non-saturable, so that sorry, liver needs an enzyme that is non-saturable, so that more like irrespective of the substrate concentration, the reaction will proceed towards the forward direction. Okay, another thing to note that uh, in hexokinase, the Km is very low for glucose. So low Km means high affinity. Okay. And for glucokinase, the Km is high for glucose. So high Km for glucokinase actually determines that when the glucose is high, when the glucose concentration is high, liver is happily phosphorylating the glucose and converting it into glucose 6 phosphate using glucokinase. which is non-saturable. But when the glucose concentration is low, then the enzyme should be able to release glucose as soon as possible. So with a low km, that release will not be faster, because low km means high binding affinity. High binding affinity, that means hexokinase won't be able to release glucose as readily as glucokinase, because glucokinase has a high km, so it has a low binding affinity. So when the concentration of glucose, when the concentration of substrate is very low, in that case it can release the substrate easily. And hexokinase is product feedback inhibited. Product feedback inhibited means the product, what is the product here? Product is glucose 6 phosphate, right? So glucose 6 phosphate, if the concentration of glucose 6 phosphate is high, it will inhibit the hexokinase enzyme, but there is no feedback inhibition in case of glucokinase. So glucokinase, no matter how much glucose 6 phosphate is present, glucokinase will keep converting glucose to glucose 6 phosphate if glucose concentration is high. If glucose concentration is low, glucokinase will immediately release the glucose from its binding sites, and the glucose can be transferred to the blood for basically normalizing the blood glucose level. So at high blood glucose concentration, liver has a net uptake of glucose. So liver will, when liver will see the sense that uh, blood glucose is high, it will uptake the glucose and proceed towards glycogenesis. And uh, but it is net producer of glucose. So when glucose concentrations are low in the blood, it will start producing the glucose by glycogenolysis, and it will basically start releasing the glucose from liver to blood. Okay, is this clear? Okay, moving on to the next question then. Which two pancreatic hormones are primarily responsible for blood glucose regulation? This is a very common question. We have been discussing this since week 1. So, which two pancreatic hormones are primarily responsible for blood glucose regulation? Option A, insulin and glucagon. Option B, insulin and somatostatin. Option C, glucagon and amylin. Option D, amylin and somatostatin. What is the correct answer? You're saying option A, that is insulin and glucagon. That is absolutely correct. So, insulin and glucagon, as we have discussed previously also in the previous sessions, insulin and glucagon both are hormones that are secreted by pancreas at different blood glucose levels. So, if the insulin is secreted, it signals the anabolic pathways where the glucose is metabolized, the glucose is converted into different substrates. Okay, and when glucagon is synthesized, then it will trigger towards glucose synthesis, that is catabolic pathway, that will trigger towards glycogen breakdown and lipid breakdown, basically all the breakdown processes that can ultimately form more and more glucose. So glucagon leads to catabolic processes, insulin leads to anabolic processes. Okay, I hope that's clear. Next question. All of these factors leading to decrease in blood glucose level except option A, oxidation of glucose by tissue to supply energy. Option B, hepatic glycogenesis. Option C, glycogen breakdown in muscles. Option D, conversion of glucose to fats in adipose tissues. Option E, formation of ribose sugar and nucleic acid synthesis. Option So all of these factors leading to decrease in blood glucose level except oxidation of glucose by tissue to supply energy, hepatic glycogenesis, glycogen breakdown in muscle, conversion of glucose to fats in adipose tissues, and formation of ribose sugars and nucleic acid synthesis. Basically, which of the following leads to increase in blood glucose instead of decrease? What would be the correct answer? You are saying option E. Think again. That is not the correct answer. So you have to see which processes are leading to glucose formation and which processes are leading to glucose breakdown. So you see in the first option, oxidation of glucose by tissue to supply energy, that is basically glycolysis. Okay. So glycolysis, here the glucose is being oxidized. So it will lead to decrease in blood glucose level, right? So it will decrease the glucose level in blood. Then we have hepatic glycogenesis. So hepatic glycogenesis basically means glycogen synthesis in liver. Okay, glycogenesis means glycogen synthesis. When does glycogen synthesis happen? When high glucose is there. So that will lead to decrease in glucose level again, because glucose will be taken up for the formation of glycogen. Then what is the third option? Glycogen breakdown in muscle. What happens in glycogen breakdown? Can you tell me what happens in glycogen breakdown? What would be the product of glycogen breakdown? It will be converted to glucose, right? So it will be converted to glucose. That means glucose level is going to increase. So this will be the correct answer. So this is not going to decrease the blood glucose level. It is going to increase the blood glucose level because glycogen will be broken down. More and more glucose will form, and that glucose will ultimately transported. That glucose will be ultimately transported to blood. The fourth option was conversion of glucose to fats and adipose tissues. Again, glucose will be taken up for the formation of fats. Okay, so this will also decrease the glucose level. Then the last was formation of ribose sugars and nucleic acid synthesis. Do you remember which pathway we are talking about when we say ribose sugar and nucleic acid synthesis? Do you remember the pathway?
It's HMP shunt, no, ma'am.
Yes, it's HMP shunt. So in HMP shunt, what was the starting material? Starting material was,
Glucose 6-phosphate.
yes. So where is the glucose 6
But
phosphate coming from? It is coming from glucose.
glycogen breakdown in muscles is also due to when there is low glucose, no. Then only when the glycogen storage will get breakdown, no, ma'am, in muscles.
So
But that is also due to decreased blood
that
glucose.
will that will lead, that is due to the decrease in
Okay.
blood glucose level, but it will ultimately lead to increase in blood glucose level. You understand
Okay.
the question? The
Yes,
question
yes.
is what is leading to decrease. Okay,
Hmm.
so glycogen breakdown will happen after there is a decrease in blood glucose level. Glycogen will be broken down so that the blood glucose level can be restored or can be increased. Okay, so this is a
Okay.
factor that will lead to increase. Okay, and all the other things will lead to decrease. Do you understand?
Yes, ma'am. But glucose to fat in adipose tissue also occurs when during glycolysis the glyceraldehyde phosphate is diverted for fat metabolism. No, even then when the glucose level will be either normal or it will be high. No,
No, glucose. So ultimately, glucose is taken.
okay. Hmm.
Glucose is basically used up to process to make fats in the adipose tissue. So glucose is used, so that means it is leading to the decrease in glucose. That is, so all these processes are interconnected to each other. So if you think of it in this way, that this is leading to this is leading to decrease in blood glucose, so
So
glycogen breakdown
glucose
will
is
start.
utilized. Where and all
uh,
glucose is utilized, then it
that
levels
that is
decreases.
that is the where and all glucose is being utilized, those processes are going to lead to decrease in blood glucose level. Later,
Okay.
obviously, as soon as body detects the decrease in blood glucose level, it starts the other processes that are going to increase the blood glucose level. Okay.
Okay, ma'am. Thank you.
So the correct answer here is option C. So these are the factors that leads to increase in blood glucose level, like absorption from the intestine. So once we have once we are eating the food, whatever glucose is present, it is, once it is absorbed from the intestine into the blood, it is going to increase the blood glucose level. So if you are having a carbohydrate rich diet, immediately after eating, like after within few hours of eating, you will see there is a blood glucose spike in your blood. So that spike is due to absorption of the glucose from the intestine. Then you have hepatic glycogenolysis. So glycogen, whenever glycogen is broken down in liver, it is going to increase the blood glucose level, because glycogen will lead to glucose formation that will finally transport it to blood. Then gluconeogenesis, again a reverse process of glycolysis. So gluconeogenesis leads to glucose formation. Then glucose obtained from other carbohydrates like fructose, galactose. So all these ultimately can be converted to glucose. So fructose can be converted to glucose, or galactose can be converted to glucose using some isomerase enzymes. So that is a different metabolism that can also lead to increase in glucose. Now factors that are that can decrease the blood glucose level. So first is oxidation of glucose by tissue to supply energy, that is basically glycolysis. Then hepatic glycogenesis, which is glycogen formation, glycogen synthesis. So again, glucose will be taken up to synthesize glycogen. Glycogen formation in muscle, same thing like liver glycogenesis. This is muscle glycogenesis. Then we have conversion of glucose to fats in adipose tissues. So glucose will be taken up and used to make fats in the adipose tissue. Then synthesis and formation of fructose and seminal fluid, lactose and mammary glands. In both the cases, glucose is used up for the formation of fructose or for the formation of lactose. Then you have formation of ribose sugars and nucleic acid synthesis, which is the HMP shunt pathway, as you mentioned, which starts from glucose 6 phosphate, which comes from glucose ultimately. Okay. So all the processes where glucose is the starting product, that is going to lead that is going to decrease the blood glucose level. All the process where glucose is the end product, that is going to increase the blood glucose level. Think of it like that. Okay, I hope that's clear now. Okay, so basically you are thinking in a way like what will happen if blood glucose level is low, but I am asking like what all processes can lead to low blood glucose. Okay, so glycogen breakdown will happen when blood glucose is low, but glycogen breakdown is happening because the blood glucose is low and now it wants to increase the blood glucose level. So glycogen breakdown is going to lead to increase in blood glucose level, right? So think of it in that way. Is it clear now?
After much. Yes, ma'am. Clear, ma'am. Thank you.
So this is how the homeostasis is maintained. Uh, so when the blood glucose level is high, uh, it stimulates the basically it stimulates the insulin secretion from the pancreas, beta cells of the pancreas, and that insulin will basically trigger different processes. So it will trigger liver to take up the glucose and store it at glycogen. It will trigger the muscles to use the glucose for glycolytic processes. Then blood glucose level in ultimately will go down. So basically, conversion of glucose to glycogen or conversion of glucose to uh, pyruvate and acetyl coenzyme, basically glycolytic pathways. All these are going to decrease the blood glucose level and we'll come to a normal level. If only this top part keeps on happening, what will happen? The blood glucose level will go below the normal levels. Once it goes below the normal levels, the pancreas will start secreting glucagon instead of insulin, and that glucagon is going to trigger the liver to break down the glycogen. So whatever glycogen is stored here, now it will trigger the liver to break down the glycogen to release glucose, and then glucose levels will be again high, and basically glycogen breakdown is now leading to increase in glucose levels till it reaches a normal. So there is a normal blood glucose level that has to be maintained in the body. If it goes high, our body has certain processes to bring it back to the normal. If it goes low, then also our body has certain processes to bring it back to the normal. Okay, we'll move on to the next question now. Which of the following is an autoimmune disease that results in the destruction of insulin producing beta cells in the pancreas? Which of the following is an autoimmune disease that results in the destruction of insulin producing beta cells in the pancreas? Option A, type 1 diabetes. Option B, type 2 diabetes. Option C, gestational diabetes. Option D, maturity onset diabetes of the young. Which of the following is an autoimmune disease that results in the destruction of insulin producing beta cells in the pancreas? So, option A, you are saying that is type 1 diabetes. That is the correct answer. So, type 1 diabetes is an autoimmune disease where it results to, so the immune system of the body starts killing the beta cells of the pancreas. That leads to uh, that leads to failure in the insulin production. So insulin is not produced in the body, and then due to absence of insulin, blood glucose level keeps increasing. So if you remember from this diagram, if the insulin is not present, it can't trigger the liver to convert the glucose into glycogen, and blood glucose level will keep on increasing, and that's why a patient becomes diabetic. Okay, so the major difference between type 1 and type 2 diabetes is this only, that in type 1 diabetes the beta cells are completely destroyed by the immune system, so it's an autoimmune disorder where beta cells are not present, so there's no insulin at all, there's no insulin production at all, and type 1 diabetes treatment needs the insulin injection ultimately. For type 2 diabetes, however, it's not like the insulin production has stopped completely. There is some or the other defect in the insulin production that leads to deficiency in insulin, and that leads to uh, like that gradually will lead to complete absence of insulin. So initially in type 2 diabetes, since it's due to multiple other factors, it is not completely destruction of beta cells. So there are beta cells present in the pancreas, they are just not able to produce enough insulin that is required by the body. Okay, so that's why they say that so type 2 diabetes is most common. So all the people around you, if you have ever heard that they have sugar, like it's a very common term that we use in India, like we have sugar or we have diabetes. So most of those people have type 2 diabetes. That is the more common form of diabetes. Type 1 diabetes is an autoimmune disease. It's rarer as compared to type 2 diabetes. Type 2 diabetes can be reversed by lifestyle changes, but type 1 diabetes uh, is not possible to be reversed. Type 2 diabetes is generally hereditary also. So that is the major difference between type 1 and type 2 diabetes. That's clear. Okay, moving on to the next question. Major metabolic changes in diabetes mellitus includes all except option A, increase in glycogenolysis, option B, increase in gluconeogenesis, option C, increase in production of ketone bodies, option D, increase in protein synthesis, and option E, lipolysis. Major metabolic changes in diabetes mellitus includes all except. So, which of the following is not included in the major metabolic changes during diabetes mellitus? High glyco high glycogenolysis, high gluconeogenesis, high production of ketone bodies, high protein synthesis, and high lipolysis. What would be the correct answer? You are saying option D, that is protein synthesis. Yes, that is the correct answer. So during diabetes, as we have discussed, that it leads to high blood glucose level. So now high blood glucose level will be due to what all factors. So we have discussed the factors that leads to high glucose levels. So glycogenolysis is going to lead to increase in blood glucose level because glycogen is broken down into glucose. Same thing goes for gluconeogenesis. Glucose is the end product, so it is going to increase the blood glucose. Then production of ketone bodies also is a major metabolic changes. We'll discuss about ketone bodies later. So protein synthesis will use the glucose because protein synthesis will require amino acid synthesis, and amino acid synthesis basically uh, comes from different byproducts of the glucose metabolic pathway. So it requires glucose, whereas protein breakdown can actually lead to glucose formation. So protein breakdown is a metabolic change that happens in diabetes mellitus. Then lipolysis, again it's breakdown of lipids, that is also going to lead to formation of more and more glucose. So protein synthesis is the correct answer. So these are the metabolic changes that occurs during diabetes mellitus. So it is divided into three different metabolic pathways. So one is CHO metabolism, one is lipid metabolism, and one is protein metabolism. So in CHO metabolism, glucose uptake is reduced because glucose is going to increase in the blood, right? So glucose uptake is reduced, that's why glucose is high in the blood. Then high glycogenolysis, that is high glycogen breakdown, that's why glucose is high in the blood. Then gluconeogenesis is high, that is also leading to high blood glucose. Then in lipid metabolism, lipolysis, that is lipid breakdown, is higher, that leads to high blood glucose. Fatty acid oxidation is higher. Fatty acid oxidation leads to acetyl coenzyme formation that can enter the gluconeogenesis pathway. So that can also lead to high blood glucose. Then production of ketone bodies in liver. So more and more acetyl coenzyme A can lead to formation of these ketone bodies. Uh, which is also a major metabolic change in diabetes mellitus. Then we have protein. In protein metabolism, obviously protein synthesis is down and protein degradation increases. That can lead to that can enter this gluconeogenesis pathway in some of these, in one of these steps. So that can also lead to high blood glucose. Understood? Okay. Okay. Leading to moving on to the next question. Bile salts aid in the digestion and absorption of lipids by. Select ones that are correct. Bile salts aid in the digestion and absorption of lipids by. So more than one option could be correct. Okay. Option A, emulsifying dietary fats. Option B, breaking down triglycerides into monoglycerides and fatty acids. Option C, enhances the pancreatic lipase activity by combining with colipase. Option D, inhibiting the activity of pancreatic lipase. Bile salts aid in the digestion and absorption of lipid by. Option A, emulsifying the dietary fats. Option B, breaking down the triglycerides and into monoglycerides and fatty acids. Option C, enhances the pancreatic lipase activity by combining with colipase. And option D, inhibiting the activity of pancreatic lipase. You're saying option A and option C. That is emulsifying dietary fats and enhancing the pancreatic lipase activity by combining with colipase. That is the correct answer. Option A and C are the correct answers. So bile salts basically, so since lipids are not water soluble, they are fats, they are not water soluble. It's like oil in water, right? So we need to make emulsions. So to dissolve oil in water also, if you would have studied, if you recall your previous science classes from school, you have studied about emulsions, that if you want to mix oil and water, you have to make emulsion of that. Okay, so same concept goes here. uh, to emulsify these lipids, these fat molecules inside the body, since our body is an aqueous system, right? All the enzymes and all work in an aqueous environment. So the lipids have to dissolve in the in this aqueous environment. So by bile salts basically help the fats to emulsify. How? They basically have a hydrophilic and a hydrophobic side, so they'll cover the fat droplets or fat globules. In a way, detergents form micelles. So if you remember the micelle formation by detergent, they will also have a hydrophobic end and hydrophilic end. So hydrophilic end will face towards the outer part and hydrophobic end will be towards the inner core. So similarly, the fat globule is the uh, hydrophobic part, that is towards the inner part, and the bile salts are covering it, exposing the hydrophilic side to in on the outer part. That will lead to solubilization of these fat globules in the aqueous environment. Okay, so it breaks down the fat globule, the bigger fat globule, into smaller globules and encapsulates it, so that it can be stabilized, it can be solubilized in the aqueous environment. So it stabilizes the lipid particles by making them smaller, and and prevent them from coalescing. So basically it prevents them from aggregating with each other and again forming the big fat globule. Okay, so this bile salt, it is synthesized in liver and stored in gallbladder. They are basically conjugation of bile acids with taurine or glycine, and they have this amphipathic nature. So amphipathic nature means it has a hydrophilic and a hydrophobic end. So it has both hydrophilic and hydrophobic nature, and it functions as detergent, as I already mentioned, and it stabilizes these lipid particles. Okay, So this is one function of bile salts. The other function is uh, to activate these pancreatic lipase. So it helps in the proper functioning of the pancreatic lipase. How? By helping in combination of lipase with a small protein called colipase. So colipase is a small protein, or you can say peptide, and you can see here this triglyceride particle, that is the fat particle. This triglyceride particle uh, will be able to metabolized by this lipase. So lipase is the enzyme that is going to break down the triglyceride particles into monoglycerides and fatty acids, right? So for lipase to act on the triglyceride, it requires the help of another protein, colipase, and this combination of colipase with the lipase, it is facilitated by these bile salts. So basically enzyme is again going to work in aqueous environment, so it requires the bile salts to be present so that the colipase can bring this triglyceride particle near the lipase catalytic site, where lipase can now act on it and break down the triglyceride into monoglyceride or glycerol and fatty acids. Okay, free fatty acids can be released. So bile salts have two functions. One is emulsification of the lipid molecules, and one is helping the pancreatic lipase. It is specifically mentioned for pancreatic lipase. Okay, so it helps the pancreatic lipase by combining it with another protein, small protein called colipase, which is a helper protein, you can say. Okay, is this clear?
Yes, ma'am, it's clear, ma'am.
Okay, we'll move to the next question now. Which of the following combination correctly matches the enzymes with their sites of action? So we have different enzymes. A lingual enzyme, B pancreatic lingual lipase, B pancreatic lipase, C hormone sensitive lipase, and D lipoprotein lipase. Okay, and then we have different locations. First is small intestine, second is capillaries, third is oral cavity, and fourth is adipose cells. So you have to match these lipases, different types of lipases, to their uh, sites of action. So where they are present, where they are uh, performing their activity. Okay, so you have four enzymes: lingual lipase, pancreatic lipase, hormone sensitive lipase, and lipoprotein lipase, and four locations: small intestine, capillaries, oral cavity, and adipose tissues. So you have to match. Option A, A1, B2, C3, D4. Option B, A2, B3, C4, D1. Option C, A3, B1, C2, D4. Option D, A3, B1, C4, D2. And option E, A4, B2, C3, and D1. What do you think is the correct answer? So you are saying option D, that is A3, B1, C4, and D2. Okay. So A3 means lingual lipase will be in oral cavity. That is correct. Then B1, that is pancreatic lipase, will act in small intestine. C4, hormone sensitive lipase, will act in adipose cells, and lipoprotein lipase will act in capillaries. Okay, so these are the different lipases and the site of action, target substrates, and products. So lingual lipase, as we discussed, it is present in the mouth. So this is the first lipase that comes in contact with the uh, food that we are eating. So whatever fat molecules are there in our food, whatever lipids are there in our food that are first digested by these lingual lipase. So all the triacyl glycerides mainly present in uh, milk. Okay, so these triacyl glycerides that are present in milk, they are like the breakdown of those triacylglycerides start in our mouth itself. So there is lingual lipase that is present in the mouth, and it is also present in the stomach. And this lipase mainly acts at a lower pH, since you know that the stomach pH is highly acidic. So these triacylglycerides and some medium chain fatty acids, they are broken down into free fatty acids and diacylglycerides. So it removes one fatty acid from the triacylglyceride to make diacylglyceride, and a free fatty acid is released. Then comes the pancreatic lipase that works along with the colipase, as we have already seen. So bile salts helps this combination of colipase to pancreatic lipase, and it is present in small intestine, uh, where it breaks the triacylglycerides with uh, along with long chain fatty acids into free fatty acids and two monoacylglycerides. Okay, so triacylglycerides with long chain fatty acids. So triacylglycerides is basically glycerol with fatty acids attached to it. Okay, now that length of fatty acid could be anything. So triacylglycerides with medium chain fatty acids are broken down by lingual lipase, but with long chain fatty acids, it can be broken down by pancreatic lipase to give free fatty acids and monoacylglycerides. Then we have intestinal lipase with bile acids, which is also present in small intestine, but it can break down only triacylglycerides with triacylglycerides with medium chain fatty acids. So you see, pancreatic lipase is the only lipase that can break down glycerides with long chain fatty acids. Okay, intestinal lipase is also present in small intestine, but it cannot break the long chain fatty acids. It will also lead to the formation of free fatty acid along with glycerol. So it will completely remove the fatty acids and leave a glycerol molecule. Then comes the phospholipase A2 plus bile salts. They are also present in small intestine, but they are present to uh, break the phospholipids with unsaturated fatty acids at position 2. So you see, all these lipases are present in different sites, and they have different target substrates. So in small intestine itself, there are three types of lipases: pancreatic lipase, intestinal lipase, and phospholipase. So pancreatic lipase along with colipase, it breaks down the glycerides with long chain fatty acids. Intestinal lipase with bile acids, they break down the triacylglycerides with medium chain fatty acids. And phospholipase A2, it breaks down the phospholipids with unsaturated fatty acids in the small intestine. So in the small intestine itself, it has three different types of lipases, and the product is unsaturated free fatty acids and lysolecithin. Then we have lipoprotein lipase that is present in the capillary walls. It leads to the formation of triacylglycerides and chylomicron or VLDL. We'll discuss this chylomicron in detail. So lipoprotein lipase, they are present in capillary walls, and they also lead to the formation of free fatty acid and glycerol. Then we have hormone sensitive lipase that is present in adipose cells, and that can break down triacylglycerides stored in the adipose cells, and it can also lead to the formation of free fatty acid and glycerol. Okay. Moving on to the next question. Which of the following molecule acts as the primary transporter for absorbed lipids in the intestine? Which molecule acts as the primary transporter for absorbed lipid in the intestine? Option A, chylomicron. Option B, VLDL. Option C, HDL. Option D, LDL. Which molecule acts as a primary transporter for absorbed lipids in the intestine? You are saying option A, chylomicron. That is the correct answer. So chylomicrons are basically uh, carrier molecules that carry all types of triacylglycerides and cholesterol and phospholipids. So whatever uh, uh, fatty acids or lipids are present, they are first broken down and then again reassembled. After the reassembly, they are combined in a they are carried by a molecule called chylomicron. So the chylomicron is a collection of all these triacylglycerides and cholesterol, all types of lipid molecules, along with some protein. So they form a structure that can be absorbed by the intestinal tract, that can be transported from the intestinal tract via the lymphatic vessels, and later it enters the thoracic duct, where it can be transported to the peripheral capillaries, where again the digestion can happen. It can be broken down into different fatty acids and monoglycerides, which can be absorbed by the liver, adipose cells, cardiac muscles, skeletal muscles, and etc. Okay, so chylomicron is a structure that basically a transporter molecule that transports all these lipids to from the intestine to the blood, and finally to the destination, all the different cells, through the lymphatic vessels, and then finally to the peripheral capillaries. Okay, this is clear. Okay, moving on to the next question. A baby with galactosemia should avoid which food? A baby with galactosemia should avoid which food? Option A, rice. Option B, cow's milk. Option C, chicken. Option D, eggs. A baby with galactosemia should avoid which food? You are saying option B, cow's milk. That is the correct answer. So, galactosemia, if you remember from the first slides, galactosemia is due to the deficiency of uh, uridyl transferase, right? Galactose 1 phosphate uridyl transferase. So, basically, it is a defect in the galactose metabolism, right? It is a defect in the galactose metabolism. Now, if you see in cow's milk will contain lactose. Lactose is a disaccharide made of glucose and galactose. So lactose will be broken down into glucose and galactose. Okay. So if there is a defect in galactose metabolism, this galactose won't be able to process, and then galactose will the accumulation of galactose will ultimately lead to hypoglycemia and other uh, metabolic uh, disorders like glycosuria and all. So galactosemia, if a patient has galactosemia, if a baby has galactosemia, it should avoid things that has lactose in it or that has galactose in it. Okay, so lactose will ultimately be broken down into glucose and galactose, as it is a disaccharide made from glucose and galactose. So if lactose is present in the diet, it will uh, lead to the formation of galactose, and since there's galactosemia, that is there's a deficiency in an enzyme that is present that is required in the galactose metabolism. This galactose won't be metabolized, this galactose will be accumulated, and it will lead to multiple disorders. Okay, so a baby with galactosemia should avoid food with lactose in it or food with galactose in it. That's clear. Okay, moving to the next question. Essential fructosuria results from a deficiency of option A aldolase B, option B fructokinase, option C hexokinase, and option D phosphofructokinase. Essential fructosuria results from the deficiency of aldolase B, fructokinase, hexokinase, and phosphofructokinase. You are saying option B, that is fructokinase. That is the correct answer. So, so if you remember the fructose metabolism, the fructose is first converted to fructose 1 phosphate by fructokinase, and then fructose 1 phosphate is converted to dihydroxyacetone phosphate and glyceraldehyde by aldolase B. So this absence of aldolase B we studied, it leads to hereditary fructose intolerance. Absence of fructokinase leads to fructosuria. So absence of fructokinase will ultimately lead to accumulation of fructose. Accumulation of fructose will lead to the excretion of fructose in the urine, leading to fructosuria. Okay, so it's a benign metabolic defect. It doesn't really cause any abnormality. Only fructose is really excreted in the urine. So that is the main symptom or sign that can be like that is detected. Okay, so it's a benign metabolic defect due to the deficiency of hepatic fructokinase. So fructokinase is not present in liver. Thus, fructose keeps getting accumulated and finally excreted in the urine. So the incidence is 1 in 130,000 births. So it's quite rare. Fructose is not converted to fructose 1 phosphate. There is no abnormality other than excretion of fructose in urine. Treatment is not indicated, and urine gives positive Benedict's and Selvinov's test. So, Benedict's test is generally done for reducing sugar. Fructose is a reducing sugar. Glucose, fructose, all of these are reducing sugar. So, it will give a positive Benedict's test if fructose is present in the urine. Okay. So, that is the fructosuria. Uh, like fructosuria occurs due to the deficiency of fructokinase. Is that clear? Glucagon acts directly on which organ to raise blood glucose? Glucagon acts directly on which organ to raise blood glucose? Option A kidney, option B liver, option C muscle, and option D adipose tissue. Glucagon acts directly on which organ to raise the blood glucose? You're saying option B, that is liver. That is the correct answer. So, glucagon acts on liver and it triggers the uh, gluconeogenesis and glycogenolysis pathways where more and more blood glucose can be formed, can be increased. Okay, so glucagon and insulin, all these uh, are the hormones that are secreted by pancreas and they are main regulators of blood glucose levels. Okay, which condition is a common acute complication of type 1 diabetes? Which condition is a common acute complication of type 1 diabetes? Option A diabetic nephropathy, option B hypertension, option C diabetic ketoacidosis, and option D atherosclerosis. So you have to tell the acute complication that is present only in type 1 diabetes or major in type 1 diabetes. Okay, diabetic nephropathy, hypertension, diabetic ketoacidosis, and atherosclerosis. So, what is the correct answer here? You're saying option C, that is diabetic ketoacidosis. That is the correct answer. So, this is basically the formation of ketone bodies, and this is more prevalent in type 1 diabetes than type 2 diabetes. So, in type 1 diabetes, as you know, it's an autoimmune disorder where the beta cells of pancreas that secretes insulin are destroyed. So as a result, there is no insulin present in secretion and only glucagon is present. Now glucagon is inhibited by insulin, but since there is no insulin, there is no inhibition of glucagon, and more glucagon will lead to all the pathways that will lead to glucose formation. So you can see glucagon will trigger the gluconeogenesis, where pyruvate is converted to glucose 6 phosphate and glucose, which is transported out of the liver into the blood. Then this pyruvate will also get converted to acetyl coenzyme A, but then acetyl coenzyme A won't be able to enter TCA cycle after a point because there will be lack of oxaloacetate, the substrate of TCA cycle. So in the absence of oxaloacetate, acetyl coenzyme A will not be able to enter the TCA cycle. As a result, acetyl coenzyme A will lead to formation of something called ketone bodies, and these ketone bodies will be released in the blood. Okay, and this ketosis results from the massive mobilization of fatty acids from the adipose, from followed by hepatic ketogenesis. So basically, high acetyl coenzyme is coming from the fatty acid metabolism. So when you will study during fatty acid oxidation, ultimate end product is acetyl coenzyme. So you see, uh, from the triacyl glycerides, we were getting fatty acids and glycerols, right? Now, this glycerol will be converted to gluconeogenic precursors and pyruvate. Pyruvate can either go to towards glucose or towards acetyl coenzyme. Then we have fatty acids that is also going to go towards acetyl coenzyme. Then we have other, so in the liver, these fatty acids can again be converted to triacylglycerol. Those triacylglycerol will lead to VLDLs that will also keep getting accumulated in the blood. Then other amino acids and uh, whatever amino acids are present in the muscle and other peripheral tissue, they will also enter the gluconeogenic, they will also become the gluconeogenic precursors, and they will also lead to the formation of pyruvate that will go towards the glucose formation. Then glycogen will obviously be broken down into glucose formation. So glucose level is increasing in the blood along with the acetyl coenzyme concentration that is increasing in the liver, leading to the formation of ketone bodies that will also be released in the blood. So ketone body concentration will be increased. So that is called the hepatic ketogenesis or uh, ketosis. Then VLDLs will also be accumulated in the blood, and chylomicrons are accumulated in the blood. So type 1 diabetes have different effects. So high blood glucose level obviously is going to lead to hyperglycemia. It is resulting from the increased gluconeogenesis and decreased glucose uptake in the peripheral tissue. Okay, there's no insulin to trigger the glucose uptake, so more and more glucose is formed, but there's no uh, trigger to signal the uptake of glucose. So that's why the blood glucose level is high. Okay, so this Ketoacidosis, it's a very typical feature of type 1 diabetes. In type 2 diabetes, ketone bodies form, but they are lesser as compared to type 1 diabetes. Okay. In type 2 diabetes, which of the following is primarily impaired? Insulin secretion, option B, glycogen breakdown, option C, glucose absorption, and option D, insulin sensitivity. In type 2 diabetes, which of the following is primarily impaired? Insulin secretion, glycogen breakdown, glucose absorption, and insulin sensitivity. What is primarily impaired? What is not present in type 2 diabetes? You are saying option C, that is glucose absorption. Okay, the correct answer here actually is option D, insulin sensitivity. So, glucose absorption is due to the insulin sensitivity. So, insulin uh, is not secreted up to the point it should be secreted. Okay, so in type 1 diabetes, insulin secretion is completely impaired. Okay, so uh, insulin, there is no insulin secretion at all. But in type 2 diabetes, the insulin secretion is sensitive. So there is some level of insulin secretion at the initial stages of type 2 diabetes, but it is not sufficient to meet the body's requirement. So glucose absorption is affected due to the insulin sensitivity. But uh, what is primarily impaired in the type 2 diabetes is the insulin sensitivity. Okay, is that clear to you? Okay. Okay, so moving on to the next question then. What is the main reason for cataract formation in untreated galactosemia? What is the main reason for cataract formation in untreated galactosemia? Option A, low blood glucose. Option B, accumulation of galactitol in the lens. Option C, increased protein synthesis. And option D, vitamin D deficiency. What is the main reason for cataract formation in untreated galactosemia? Low blood glucose, accumulation of galactitol in the lens, increased protein synthesis, and vitamin D deficiency. You are saying option B, that is accumulation of galactitol in the lens. That is the correct answer. So, basically, if galactosemia is not treated, galactose is going to get accumulated, and that galactose is then converted to galactitol by uh, aldose reductase. So there are some aldose enzymes that can reduce reduce this aldehyde to an alcohol form. So galactose can become galactitol, that is the alcohol. So this, you can see this aldehyde group CHO, it is converted to CH2OH. So uh, this reduction of alcohol aldehyde to alcohol, uh, this is carried out by aldose reductase, and this alcohol form of this sugar, uh, it leads to osmotic effects that contribute to the injury of lens proteins and development of cataracts. So this is, if galactosemia is not treated, galactose accumulation will increase, and then accumulated galactose will be converted to galactitol, that is the alcohol form of a sugar, and this sugar alcohol will lead to osmotic effects uh, it will basically create an osmotic pressure which will contribute to the injury of lens proteins and development of cataracts. Okay, so there's another like similar mechanism that happens during fructose metabolism also. So in case of fructose metabolic disorders where fructose is getting accumulated, it also gets converted to alcohol form of fructose, or in case of glucose also. So glucose uh, and fructose, they can also be converted to alcohol form. So in case of diabetes, you must have heard that it affects the kidney and eyes. uh, do you know what is the alcohol form that is that forms from glucose in case of diabetes? There also an aldose enzyme acts, which converts the alcohol group of the glucose to aldehyde. Do you know the name of the alcohol? Yes, sorbitol. That is correct. So basically, glucose gets converted to sorbitol, and this sorbitol again creates osmotic imbalances in the cells, leading to uh, different symptoms that we observe during diabetes. Okay, so high glucose level gets converted to sorbitol, and this alcohol, sugar alcohol, it leads to cataract and it leads to different kidney problems. This sorbitol can be converted to fructose by sorbitol dehydrogenase. So this sorbitol can further be converted to fructose by sorbitol dehydrogenase, that it could be reduced to fructose. But sorbitol dehydrogenase is absent in certain cells, in eyes and in kidney and some other cells. It is absent. So because of that, sorbitol accumulation occurs, and sorbitol accumulation leads to different uh, uh, physiological problems. So same like galactitol, sorbitol is another sugar alcohol that creates a lot of problems in the body. So is this clear? So this is the last question in my slide today. So do you have any question regarding all the things that have been discussed today? Everything is clear? Okay. Okay, then if there's no doubt, then I think I can end this session. So you can solve your assignment questions. All the best, and uh, we'll see we'll see each other next week. Okay, then I'm ending this session now. Thank you so much for joining. Bye bye.